This is a production of Cornell University.
All right, thank you so much for that generous introduction, Mark. It's really terrific to be able to give my exit seminar in person. If on Zoom I ever move the mic from my mouth, just type in the chat and we'll try and fix that for you real fast. So I'd love to start my talk today by sharing the motivation behind my dissertation, which was to identify ways that we can further leverage the currency of plant breeding, that being genetic variation. And then I'll introduce one of my first projects that I took on at Cornell, which was to use a causal variant approach to identifying genetic variation during the mutation case study. That inspired largely the remainder of my dissertation, where I'll introduce the topic of controlled recombination and whether as breeders we might be interested in manipulating recombination to further leverage the power of selection in plant breeding during the meiosis case study. And then there are some things that you only learn in retrospect, and I'll conclude on some lessons that I've learned during my PhD. So plant breeders rely on heritable genetic diversity and variation for trait improvement. And two of the primary sources of genetic variation are recombination and mutation of genetic material during meioses. And the crossovers that occur during meioses generate the novel and favorable allelic combinations that breeders can select upon. However, these crossovers are very tightly regulated. This is a scaled illustration of a wheat chromosome. And we expect to see 1 to 2 crossovers per chromosome per meiosis. And then the distribution along the chromosome, we expect those 1 to 2 crossovers to fall on the chromosome arms away from the pericentromere. And so this limited frequency and biased distribution ultimately governs our response to selection and genetic gains. Another source of genetic variation are mutations. And in a population, novel mutations can continuously arise, and their effects can range from lethal to beneficial. As breeders, our ability to actually detect and select on those mutations is limited by genetic resolution via recombination. So hence this being a Venn diagram for breeders, these two independent processes can have overlapping consequences. Some additional points of overlap between these two processes, starting on the left-hand side. In those regions where there's fewer crossovers, deleterious mutations can start to accumulate over lots of generations in a breeding program and evolution, as well as on the right-hand side, through population improvement and artificial selection, the allele frequencies of some deleterious mutations can increase due to hitchhiking through selection and because they might be in linkage with traits of interest, and then we get these repulsion linkages. So given this tight relationship between recombination, mutation, and genetic resolution, as well as at the beginning of my PhD in 2017, the recent release of an annotated wheat reference genome, I sought to address two primary questions during my dissertation using wheat as a model. And the first being, how can geneticists improve the accuracy of identifying targets for positional cloning? This is the first project that I took on in Mark's lab, and the results of it largely inspired the remainder of my dissertation and asking the question, should breeders seek to manipulate recombination to further harness the power of selection? With the advancement of gene editing techniques, there are some pretty exciting proof of principle studies in which this has been achieved, changing that frequency or distribution. And I wanted to ask, thinking many years in the future, potentially for a breeding program, would we want to apply these technologies? So with that, let's jump into the mutation case study where we're thinking about a pretty dramatic form of mutation, structural variation. So I want to start by reviewing the process of causal variant validation via positional cloning. This is a foundational approach to go beyond mapping traits that can span broad genomic regions to validating selectable markers that can help drive genetic gains. And this traditional roadmap has been quite successful for diploid crops such as rice. And it generally starts with what QTL of interest in your population, pursuing a fine mapping population where we can narrow our region of interest, exploring that region of interest in the reference genome, looking for candidate genes, sequencing those candidate genes to look for variation, and then with your top candidates actually pursuing functional validation, for example, with a gene edited knockout. And like I mentioned, this has been quite successful for some diploid crops. And with the release of a fully annotated reference genome and the advancement of gene editing techniques, this has opened the door to positional cloning opportunities in wheat as well. So in the sorrels lab, we've worked with this biparental population, W7984 by Opata, for over a decade. And consistently, we've observed a QTL that has strong associations on chromosome 5A on the long arm with grain weight variation. And in 2017, when we became collaborators on the Wheat Coordinated Agricultural Project, which is a USDA grant, and the goal was to identify genes that can contribute to yield improvement, we thought that this would be a good QTL to pursue for positional cloning in collaboration with the grant. Now, when I dive into that story today, I want you to keep in mind that polyploids have a lot of genomic structural variation. And structural variation meaning you could have insertions, deletions, inversions, translocations along your chromosome. They can be small, they can be large, but in a polyploid, the other genomes can buffer those effects and your phenotype can be pretty muted, if even measurable to the breeder. So wheat has an A, a B, and a D genome. And I'm going to jump into the story now of how I tried to positionally clone the QTL on chromosome 5A on the long arm, and how it was overpowered by a structural variant, and present some recommendations to the field that I hope can help reduce the likelihood of mapping in the wrong direction and are more sensitive to these structural variants that are common in polyploids. And so, in order to find map this QTL, I took a heterogeneous inbred family approach, or abbreviated as a HIF. And the advantage of a HIF is that you start with a very inbred line. We had recombinant inbred lines available for our biparental. And we identified one that was segregating for the flanking markers of our QTL. Through lots and lots of rounds of inbreeding many of its progeny, we had thousands of recombination events that had a chance of recombining between our flanking markers of our QTL. This approach is nice because you have more confidence then in associating the phenotype with those recombination events, as you have a really homogeneous background genome. And this is advantageous for a quantitative trait like grain weight because of those subtle variations in the phenotype. So here we're starting by just looking at the positive and negative controls of the fine mapping population. The Opata alleles all the way across in purple of that chromosome arm portion of 5AL, where we had fine mapped our had started our QTL mapping process. So the positive controls, purple Opata, it's associated with a greater grain weight on the right-hand side of the graph. And then the W7984 alleles in teal are associated with our negative control, as they have the lesser grain weight. And we could then zoom out and look at all the different recombination events that we resolved in our fine mapping population. And through a process of elimination, we can start to associate the positive Opata alleles with a greater grain weight, and then genomic regions that have the W7984 teal alleles and a lesser grain weight. We validated this with t-tests as well and narrowed our QTL to a 10 megabase pair region, blanking 37 genes. Now, I knew that I would eventually want to look at the transcriptome for our candidate gene. So I also looked at the phenotypes as the grain developed after anthesis and narrowed the first phenotypic difference was detected at 10 days post-anthesis, which gave us confidence that this trait was expressed early, if it was controlled by the transcriptome, was expressed earlier in grain development. However, around the same time that we were pursuing the fine mapping population and identifying our transcriptome study, a publication came out where the authors worked with the same biparental. And one of the most relevant findings from their study was that the short arm of chromosome 5A of the W7984 parent was missing. So what this suggested is that the population was segregating for either the presence of the short arm of the Opata parent or an absence of a short arm. And we're working with the same population, looking at the same chromosome. We didn't notice any significant morphological variation or infertility, which is characteristic of a missing chromosome arm. But nonetheless, this really caught our attention and prompted us to investigate what was going on with the short arm in our population. So I began by just running some presence-absence markers on our fine mapping population. And generally, more individuals had the short arm than had it missing just based on these markers. But when we subset our population based on which allele was present at the QTL, we saw a really skewed distribution. The presence of the short arm was in tight linkage with the OPATA allele at the QTL, and the presence of, or the absence of the short arm, was in really tight linkage with the W7984 allele at the QTL. We can also look at this from the phenotypic distribution perspective. So on the y-axis here is the 1,000 grain weight distribution, and on the x-axis I've just subset based on the absence of the short arm and the presence of the short arm, and the colors are the OPATA allele, which we have been associating with that greater grain weight until this point in our study, and then the W7984 alleles associated with the lesser grain weight. So I just want to call your attention mainly to the few individuals that did break LD. On the left-hand side, for those couple individuals that are missing the short arm but have the OPATA alleles in purple, those individuals have some of the lower end of the grain weight of all the individuals that had the OPATA allele. On the right-hand side, that red star next to the one individual that has the short arm but was missing the but has the W7984 allele at the QTL has the greatest grain weight of all the W7984 alleles. So ultimately, all this suggests is the presence and absence of the short arm could be contributing to our difference in grain weight, despite our best efforts to develop a very homogeneous background genome. The QTL or the structural variant, we're not sure which one is driving the difference in grain weight. So this is where the transcriptome really came in handy. I looked at a range of different haplotypes and time points, and I'm just going to touch on a couple that are most important to the study today. I'll present the positive control, which was a haplotype linkage between the presence of the short arm and the positive variant of the QTL, and the absence of the short arm and the negative variant of the QTL. And we looked at the transcriptome genome-wide, but I'm just going to show you chromosome 5A because we were successful in that regard of a fine mapping population. We did only see differential expression on our chromosome of interest. So chromosome 5A is on the x-axis, and then differential expression is on the y-axis. So anything above that yellow line, which is our threshold for significance, means the positive and negative control for a given gene had a significant difference in expression. And the majority of the differential expression falls on that short arm. That's actually due to no expression at all from our negative control. So that validated that it was indeed missing in our population. And the next area I want to draw your attention to is highlighted in red. That's the 10 megabase pair region of the QTL that I had fine mapped. And I did detect expression for those genes from the positive and the negative control, but there was no significant difference in their expression. So this certainly made us pause and consider the numerous years that we had invested in this project and driving towards this QTL in this region. It made me think, did we get the wrong time point for expression? Or maybe this is not driven by the transcriptome, something's happening post-transcriptome or post-translational modifications. So because we had done a full RNA-seq experiment, I was able to look at the coding sequence, and we ultimately did not identify any significant difference in the coding sequence variation either. So what this ultimately starts to suggest to us is that the difference in grain weight was likely driven by this chromosome structural variation buffered by the other genomes, and the QTL was likely presenting as an artifact of very strong linkage with the presence absence of the short arm. Ultimately, this study presents a pretty important point of how do we detect chromosome structural variation in polyploids. And I was working on this project, the W7984 by OPATA populations are some of the most widely cited mapping populations up until we got our reference genome in 2017. And so aside from the very subtle phenotypic variation associated with a whole chromosome arm deletion, something that I wondered was, why did it take so long for the wider wheat community to identify this structural variant in a popular population? And I think I can explain that a little bit with a timeline of events. Up until 2011, the population had only been genotyped with SSR and DART markers. So those are scored on a presence-absence basis and wouldn't have necessarily alerted us to anything different from a structural variation presence-absence. In 2012, a GBS genetic map was developed, but again, working with the genetic map, it might not have alerted us to the fact that all the markers were mapping to the long arm. it was really in 2017 with the release of the reference genome that I think set us up to identify this structural variant. And it also is what allowed for the wheat cap project to move forward and giving positional cloning projects to grad students. When I was working on developing a lot of the fine mapping markers for this project in the early 2018, that's when I first started to notice that I was having trouble identifying site-specific polymorphisms on the short arm, but as our QTL was mapping to the long arm, it wasn't my primary concern. It was really in 2019 for Gutierrez Gonzalez et al.'s group with the conjunction of access to a physical map as well as the dense GBS mapping that identified the structural variant. And I just want to stress that we generally think of dramatic structural variations as associated with a pretty dramatic phenotype or changes in the fertility of the plant. But in a polyploid, it's important to keep in mind that the other genomes can buffer that effect. And across different lab groups, years, research projects, and environments, a structural variant as dramatic as a whole chromosome arm missing kind of flew under the radar for us. These are some other recent publications highlighted here that have, with the access to a reference genome and now more of a pan-genome approach in wheat, have underscored the structural diversity of wheat and how important it is to be mindful of structural variation when you're taking a causal variant approach to breeding. And so I've shared with you today my experience with positional cloning and a polyploid and how taking that traditional roadmap to positional cloning that I explained at the top overlooked some unique hurdles to polyploids. And I think that if grad students or research project groups are going to continue to invest in positional cloning projects for polyploids, there are some updates that we should make to that roadmap. And I'm going to make some recommendations that I hope could be implemented in the near future that would reduce the likelihood of mapping in the wrong direction. And all of those recommendations can be found in my paper. I also created a GitHub repository that is structured like a learning resource. Not only is it our full reproducibility of the paper and the supplementary material, it's very easily digestible no matter your experience with coding, and I hope can help others who I know will encounter structural variants in their polyploids with their own experimental design and overcoming those roadblocks. But I do want to highlight a couple of recommendations most relevant to what I talked about today. The first would be to move from a SNP to a haplotype-based approach to identify genetic diversity. I think we all know that a SNP can be in association with your trait of interest, but that SNP itself is probably not causal. And in a fine mapping context, that can lead to trusting SNPs that are in linkage across really broad swaths of the genome. In our case, in linkage across an entire centromere. Ultimately, this can lead to mapping in the wrong direction, which happened in our case. Using a haplotype-based approach, not only does that give you more confidence when actually identifying your target for positional cloning because you're going beyond a biparental to validate that QTL, you also have more awareness of your general diversity across the genome, whether that's structural variants on your own chromosome or outside, or sorry, in the background genome that you might want to be mindful of during your fine mapping population development. And then the next recommendation would be to use the transcriptome to narrow your list of candidate genes. If I had followed that traditional roadmap to positional cloning, the general recommendations are to just continue to inbreed and look for recombination events that further narrow your list of candidate genes, narrow it down to just a few so that you can run RT-qPCR. If we had done that, we would have just kept mapping in the wrong direction. And it was really running a whole seeing the whole genome and looking at the transcriptome that alerted us to that structural variant. And I don't think everyone's going to be so lucky to have a publication come out at the same time you're working on a project that highlights there's a structural variant you should look for. So I think one way to do that, it would be to use the transcriptome earlier on in a fine mapping population. And with that, we're going to move on to the meiosis case study, but I do want to introduce it by sharing that the mutation case study is really what inspired the rest of my dissertation. It was experiencing firsthand that reliance that we have on meiotic recombination, not only to pursue a positional cloning project and fine mapping, but just our general awareness of diversity across the genome. And so that brings me to the subject of controlled recombination. And controlled recombination is really an umbrella term that refers to a suite of tools that can be used to modify crossover frequency and or distribution. And when I'm talking, we're returning to this scaled illustration of a chromosome in wheat with its crossover patterns. When I'm talking about frequency, I'm referring to that 1 to 2 crossovers that we expect to see per chromosome per meiosis, and distribution, meaning that we expect those 1 to 2 crossovers to fall within the chromosome arms away from the pericentromere. And there's a range of approaches that have been developed for controlled recombination. Some might take a more generalist approach where you could start by modifying the recombination frequency. This might involve like a knockout of an anti-crossover gene, and generally we just see an elevated increase in recombination where we naturally see it fall along the chromosome. It's a little trickier to change the distribution because it's not exactly what the chromosome wants to do, but it has been achieved. And for example, in this paper by Pele et al. 2017, they had pretty remarkably saw a 20-fold increased recombination in the pericentromere in turnips. And then, as opposed to the generalist approach, we could take a more site-specific approach. And this is actually achieving recombination at specific base pair intervals on your chromosome. So I imagine you're thinking about generalist or site-specific. There's a range of applications this could have in your research or breeding program. And I'd recommend you read my paper if you want to learn more about those. And for the sake of today, I want you to just keep in mind that controlled recombination is a novel approach that may more efficiently harness the power of selection for plant breeding. Now, as it's a novel approach, there's a range of questions out there still as we go from going from the lab bench and model species to crop species and a breeder's field. And a couple of questions that I really like to focus on in this area are: where in the genome should we target recombination to actually see a benefit in our breeding programs? And by how much do we have to increase recombination to see a significant response in selection? And across the literature, there have been only evaluations of controlled recombination in a breeding program at this point via simulations. And across these simulations, they're pretty optimistic, whether it's different controlled recombination approaches, crops, traits, they can predict upwards of doubling genetic gains. And when I was deciding what question I wanted to focus on in this research area and reproducing the results of some of these papers, I realized that the majority of them focus on using estimated marker effects to identify where would be an optimal recombination point. And ultimately, that limits your analysis to historical recombination, so that frequency of where we generally see recombination occur. It's going to overlook lower recombining regions and where there can be some of those repulsion linkages that I had referenced at the top of the talk related to where mutations might fall. And so that's a pretty good target for controlled recombination. And I thought, what would happen if we could create a simulation that shifts recombination towards the pericentromere? Would we be able to unlock new variation, largely motivated by just trying to make our simulation studies most reflective of what the technology might be able to accomplish 5, 10, 15 years down the road for breeders? This actually raises a tricky question then of how do you test and evaluate the effect of recombination in regions of the genome where we've rarely measured it, and using like biological priors in your simulation. And I thought that deleterious variants could present a good signal for us. So these are mutations that tend to have a negative effect on fitness, and they can contribute substantially to phenotypic variation. This could be something like an early stop codon or a non-synonymous coding variant, and they can occur at higher frequencies in lower recombining regions where they haven't been able to be purged from the genome. And through artificial selection and population improvement, sometimes that can mean being fixed in repulsion with traits of interest that we do want. And so I identified a data set in which I could simulate and ask this question of what would happen if we changed the recombination distribution and frequencies and kept track of the deleterious variants. This data set came from Dr. Catherine Jordan's 2018 paper, and we were able to get 26 diverse wheat cultivars for which they had exome capture data as well as the deleterious variant annotations. And they were the paper showed that they were concentrated in the pericentromere. So the way that I kind of used this as an input to the simulation was thinking, if you just had SNP data, your pericentromere on a genetic map, it's very scrunched together. You have a lot of markers falling within the same centimorgan interval potentially. How do you actually identify which of those you might want to use in your simulation to inform QTL? I largely used these by just being able to identify, hey, there's an early stop codon in the pericentromere. That's a good biological prior to inform our QTL, when we wanted to pair QTL distribution in the pericentromere with increasing recombination there. So I'll get into a little bit more how I use that in the parameters now. Starting with just wild type recombination, here's just a histogram of the first chromosome of wheat as an example for the SNP count along that chromosome. And we worked with about 1,200 markers per chromosome and 1 Morgan, that's about 1 crossover on these, as we used only the genetic map for these simulations. And I could simulate changing the recombination frequency just by changing the scale of the genetic map. So in this case, it's 2-fold larger. That lends itself to about 2 times more recombination, but it hasn't changed the distribution. It's still going to prefer to have more recombination on the chromosome arms as opposed to the pericentromere. And we tested up to 20 times larger genetic maps. And then you can also change the recombination distribution. This was done just by changing the scale of the map in certain regions of the chromosome. So I wanted to preferentially increase recombination in the pericentromere. It's highlighted here in red under the wild type map, and then in the yellow map, it's 2-fold larger for more recombination in the pericentromere. And I kind of referenced this with my motivation for this simulation. The QTL were either selected randomly or based on whether they had a deleterious variant annotation, and in this data set that was concentrated in the red region of the highlighted pericentromere. So getting into a little bit of our expectations, given these parameters and inputs, I'm going to refer to these map types as the wild type map type in blue, where we never changed the recombination or the genetic map. The chromosome map type in gray, just meaning that we changed recombination across the entire chromosome, and the pericentromere map type in yellow, meaning that we changed recombination just in the pericentromere. And I expected that the chromosome map type would do the best just because it's the largest map and it has the most opportunities for more recombination, and that the pericentromere map type would perform really well when it was paired with the deleterious variant annotations because you're increasing recombination where the majority of your QTL are. So let's see if this actually happened. We're looking at the y-axis. On the y-axis here is genetic variation, and it's just graphing how genetic variation depletes in our populations over 10 cycles of genomic selection. For our populations on the left-hand side is the 2-fold increased recombination maps, and on the right-hand side are the 20-fold increased recombination maps. Our map types, again, are highlighted by their colors, with blue for the wild type, the yellow for the pericentromere map type, and the gray for the chromosome map type. And on the 2-fold increased map type, I'll introduce that under increased recombination, we expected to see that a larger map would retain more genetic variation over cycles of selection compared to wild type recombination. I highlighted it in red on the 2x increased map because that's not what we saw. Around 5 or 6 cycles of genomic selection, I'm highlighting here because that's traditionally kind of our duration of a breeding program. We didn't see any real significant difference in the genetic variation retained under increased recombination. However, under 20-fold increased recombination, those larger map types did hold on to more genetic variation compared to wild type. When we consider from the same population and its response with genetic gain over 10 cycles of genomic selection, under the 2-fold increased map type, we didn't see a significant difference in genetic variation retained, so it's no surprise that they perform pretty similarly with genetic gain. But then the 20x increased map type, surprisingly, the increased maps actually have worse genetic gain predicted than the wild type, even though they had more genetic variants to be selecting from. And it turns out that the larger genetic maps had lower genomic prediction accuracy compared to wild type because under increased recombination, that LD decays faster between the markers and the QTL. So through these simulations, this trend started to emerge that there was a trade-off in the genetic variation retained under increased recombination and whether that actually would translate to an increase in genetic gain. Variation could be generated from breaking up repulsion linkages, which is beneficial and what I was intending to have happen in my simulations. And then you could also have variation retained because you had lower prediction accuracy. So I'm just going to present a few more simulation parameters that either I thought would help me understand this trade-off or actually did help us understand this trade-off and what could lead to greater genetic gains under increased recombination. So the first being the QTL annotation and whether those were deleterious variants or just randomly selected. It turns out that there's a lot of other variables that need to be in place for these to start to have a response in selection. They weren't as important as I thought they would be. But when we do have those other variables in place, which I'll get to in a moment, it is about what you'd expect, that if your QTL are in the region that you're increasing recombination, those will do better. Something that did really make a big impact was the QTL effect ratio. So we modeled different ratios of coupling or repulsion between our QTL. Coupling between your QTL, meaning two QTL that are next to each other and they are in linkage and they both have the alleles that were of interest to us. We don't really want recombination to happen there. It would be not beneficial. Under a repulsion scenario, it's the opposite. Two QTL that are in linkage, one has the allele that you want and one has the allele that you don't want, and you need recombination to get the beneficial combination. What I want to point out here is that under coupling and random scenarios, we actually didn't see very much of a penalty under increased recombination, generally performed similar to wild type, but we only saw an increase in genetic gain and a response when we had repulsion. The relationship matrix refers to how we estimated the marker effects, and the genome-wide relationship matrix is standard practice. This was not a simulation, it's all we would have access to. Basically, this is what suffers from the LD decay between the markers and the QTL under increased recombination. The causal variant relationship matrix was something unique that we could do with a simulation because it's basically just, we knew the exact locations of our QTL, and so we could feed those right into the model. And that's a proxy for perfect LD, or maybe you got really good at positional cloning and you knew the locations of all your QTL. So let's see how these different variables play together in a response to genetic gain. We're going to consider just 20 times increased recombination frequency because we didn't see a significant response under lower frequencies. And this is pretty similar to the literature. Folks have found that you need a dramatic increase in recombination to see a response in these simulation studies. 200 QTL per chromosome, so a quantitative trait with relatively high heritability because we did not see a response to selection under lower heritabilities. And then I'll present also the QTL effect ratios with different shapes on the graph. So the coupling are represented with circles and the repulsion scenarios are represented with triangles. The genome-wide relationship matrix results are first consider on the left-hand side, and then the causal variant relationship matrix are on the right-hand side. And again, this is the genetic gain on the y-axis over the 10 cycles of genomic selection. So starting with the genome-wide relationship matrix, even under 20 times increased recombination with a quantitative trait and high heritability and all of your QTL and repulsion, we didn't see necessarily that both increased genetic maps perform better than wild type. The pericentromere map type did better than wild type, but the chromosome map type, I think, just gets too large to overcome that LD decay. So we can consider then when we remove LD from the equation, the causal variant relationship matrix, LD decay from the equation. After 10 cycles of genomic selection, we can see, especially when the QTL are under repulsion, up to 15% greater genetic gains with an increased map compared to wild type recombination. So some observations that come from these simulations are that increased recombination is going to retain more genetic variation, but at least in our data set, there were some pretty limited instances in which this actually translated to greater genetic gains. If you have a quantitative trait with high heritability and really well-defined QTL locations, which I'm not familiar of a trait like that in wheat, but if we pretend that that exists in the future, having those in repulsion is going to be, those QTL and repulsion in your population would be important, and you may need to go beyond 6 cycles of genomic selection. So this is probably a deal breaker for most wheat breeders, but I do think controlled recombination is a really interesting technology and in theory has some pretty clear applications to breeding programs. based on our data and simulation inputs, I'm not convinced that a generalist approach over many cycles of selection necessarily has the right home in wheat. Across different simulation studies that have been published, there's been shown to be a variation in the response to the efficiency of controlled recombination across different species. So this could be a species-specific instance, but I have not seen results as pessimistic as what I observed in wheat published yet. I mentioned the different approaches, generalist versus site-specific. Potentially a site-specific approach for a qualitative trait could be a good application of controlled recombination in wheat. And as the technology affordability and feasibility aligns, I hope that this simulation can inform future studies as we consider what might be the right applications of controlled recombination to plant breeding. And with that, I'm going to start to wrap things up with some of those that, I only learned in retrospect during my PhD. The first is to not get too attached to your hypothesis. I think it's good to get comfortable with null or negative results. Your goal is to learn from your data and be adaptable and ask the next question. And I think the faster you learn this, the better scientist you'll be. I definitely was confronted with this for the positional cloning project. Being on a large grant where the goal was for all the groups to be cloning genes that contribute to yield components, eventually we had to recognize that the structural variant was pretty important to our project and pivot. So that took, letting go of an original hypothesis. The next would be to test your assumptions, which is another way of saying you should avoid assumptions if you can. This came up for me a lot with the controlled recombination simulations, just based on population and quantitative genetic theories that like Mueller's ratchet or the Hill-Robertson effect, we would think that increased recombination is generally beneficial. Also, a lot of the publications out there for controlled recombination really tout its benefits. So a lot of this simulation for me and the parameters that I presented were introduced as I started to realize I wasn't seeing a response to selection and I was making assumptions about the population and what actually, until we came down to the variable that prediction accuracy and LD decay was regulating a lot of the response we saw in selection. A third recommendation is as graduate students to begin early in prioritizing your data management and reproducibility best practices. I took this class in the natural resources department and I would recommend anyone take it. It really changed the way that I code and collaborate, and it'll save you when you have a bug in your code for sure. But I think one of the greater benefits of it too was the confidence to publish null or negative results and be able to validate those with really good supplementary materials. For example, I was able to do that with the positional cloning study, and it'll come up again for me with the controlled recombination simulations when those get ready to publish. And then my last recommendation is to take time to do an internship. This is one of the reasons that I haven't been on campus. I took 6 months to work for Bayer as a data scientist, and it was one of the best decisions I made in grad school. It's a really good way to grow your network and explore career opportunities. And if grad students or PIs have questions about how you can pull that off, you can always get in touch with me. And with that, I have a lot of people I'd like to thank. Mark, thank you for letting me be as independent and trusting me as you have and be able to explore the questions that I was just most inspired by over the last 5 years. To my committee, Margaret and Adam, your advice and encouragement has been so important, and your flexibility I've appreciated so much as I've navigated the last 5 years. Jean-Luc, working with you on this last chapter has made me grow as a scientist, I think, in more ways than you know, and working with your lab has really influenced the way that I present scientific information and communicate. I'm so thankful to Synapsis for the career-long relationships that we started growing here at Cornell. Thank you to the WheatCap for your funding and putting me through grad school. And to my family and friends and my parents, I couldn't have done it without you either. Thank you for always being one phone call away. And with that, thank you to all of you for your attention. I'd be happy to answer any questions.
All right, we have a little time for questions. I want to recognize Ellie's parents. They came all the way from Seattle to celebrate this event. And so anybody that has questions, I think Moira is monitoring the chat. Let us know.
Travis, what's up?
Well, on your simulations, how did you develop the marker estimates or the effect size estimates that you used for that controlled recombination study?
Definitely. Okay, so for people on Zoom, the question was in the controlled recombination simulations, how did we estimate the marker effects and the effect sizes? So it was really important to us that across the simulations, we had everything begin like at the beginning of the simulation, standardized everyone to the same variance. So that was standardized a variance of 1. The estimated marker effects came from RR-BLEP, and the effect size, the sign of the effects was largely what we had to manipulate for the different repulsion and coupling scenarios to occur. So that, sorry, I've told you this in a funny order. Once we identified our coupling and repulsion scenarios based on the QTL effect sign, then we standardized the variance that changed the effect sizes. But I did also look at the data. It's in some supplementary data from actually how different QTL effect sizes responded to selection. We saw some interesting situations where, for example, like a very, some of the smaller effect sizes, deleterious variance would still come along through selection. So that's sort of that hitchhiking effect of if it has a small enough effect, it will still stick around under increased recombination. Does that answer your question?
Yeah, a quick follow-up would be what proportion of variance came from the centromere versus the arms?
So for people on Zoom, the question is what proportion of the variance came from the centromere versus the arms? And I did not look at that. We had the markers, we had worked with 1,200 markers that were fairly evenly distributed across the chromosome. So I would say, in the case with the randomly distributed QTL, I would assume that I would have to check, but I'd assume there wasn't very much difference between the arms and the centromere, but under the deleterious variant approach where we prioritized QTL that we knew fell in those regions, there would be more variance explained by the centromere, pericentromere. Okay, thanks.
Ed's ready to ask a question, please, and let me know.
Ed, go ahead.
Hi. Well, first off, just wonderful presentation and talk there. What I was wondering about, when you knew the causal variance, did you have an a priori estimate of the effect size of the causal variant? I mean, that's one of the things that Guillaume Ramstein, when he's looked at that, getting some estimate there helped out in prediction accuracy.
That's a great question. I did not prioritize specifically certain QTL to have more of an effect size than others. I worked with AlphaSimR for this, and I just used the distribution of effect sizes that it gave. So some would be larger, smaller, but I think something I really learned from this project was having been someone who has a lab background and then transitioning later throughout my degree into more of a quantitative and data science background. You can really quickly on a computer add another variable, and then that adds a lot of work to your analysis. So it's a great question, and it's something I'd want to explore, but I think it didn't fall into one of our simulation parameters that we prioritized in our first round of analysis. Thank you.
That sounds good. Yeah, yeah. So I think Guillaume's kind of saw weighting did matter. If you could prioritize the causal variance by the predicted deleteriousness.
That's a great suggestion. I'll definitely think about how we could incorporate that. Thank you. Mike.
Very nice presentation. Just a question on, in terms of like the 20x recombination scenario, where you probably like first training the model, and then you may even have changing in phase between coupling and repulsion as generations go on. So would that then argue you would have to retrain more often for it to be effective then?
That is a great question. Okay, so for people on Zoom, the question was when you increased, like how did we retrain the model in our simulations? And that is another, point of hesitancy I have about my controlled recombination based on our simulations. I retrained it every generation
You did.
because because of our lowered prediction accuracy, and I was not seeing that response to selection. Some papers out there have actually modeled changing at how often they trained the model. But we settled on training every generation, which is not ideal for a breeding program.
But I think for 20x recombination, you would have to do it.
Yes, yeah. So under increased recombination, retraining more often is definitely or would be necessary, and if this was applied in the real world. Yeah, thank you.
I have a question.
Yes.
Looking back, gene editing was available to you for positional cloning. Would you use it in that situation?
Okay, so looking back, the question was at the positional cloning project if gene editing was available or controlled recombination. If controlled recombination was available, like from a site-specific approach, would I have used it? Well, I did try to do that. If we think back to pre-pandemic times, I would go across to the transformation facility a lot, trying to optimize conditions for doing transformations in wheat. And I had this lofty idea that I would validate my positional cloning project with doing it with far fewer individuals and gene editing. I have some hesitancies there just because we encountered, as you probably recall, how hard it was for our cultivars to take in a transformation facility. So that's another, point of consideration with controlled recombination. Something that in the future we may have more cultivars that work with tissue culture, but it was a limiting factor for me just to begin this project. Was just the biparental we were working with wasn't taking very well the tissue culture. So in an ideal world, if it was working, I think that'd be a great experiment to show that you could do positional cloning much faster and with fewer individuals using controlled recombination. And it has been shown to work in yeast, some proof of principle studies like that, but yeast is really different from plants.
Anything else?
Okay.
Let's thank Ellie for a fantastic.
Bill.
This has been a production of Cornell University, on the web at cornell.edu.